Hello everyone, my name is VASU and I am from Polytechnic Nilai Negeri Sembilan. My matrix number is 23DBT 20F 2019 and I am from DBT 3A. I am here today to talk about restriction enzyme digestion for my DYB 30093 molecular biology subject. What is restriction enzyme digestion? Restriction digestion, also known as restriction endonuclease, is a method of cutting DNA at specific locations determined by the surrounding DNA sequence. Restriction digestion is carried out by incubating the target DNA molecule with restriction enzymes, which identify and bind certain DNA sequences and cleave specific nucleotides either within or outside of the recognition sequence. Restriction digestion can produce blunt ends, which are DNA molecule ends that finish in a base pair, or sticky ends, which are DNA molecule ends that ends with a nucleotide overhang. Restriction digestion is commonly used to prepare a DNA fragment for subsequent molecular cloning because it allows DNA fragments to be pierced together. Gel electrophoresis, in which the products of the digestion are separated by molecule length in a polymer gel to which an electric field has been applied, can be used to evaluate the result of a restriction digestion. A typical restriction digestion reaction includes the DNA template, the restriction enzyme of choice, a buffer, or occasionally BSA protein. The reaction is initiated at a specific temperature required for optimal restriction enzyme activity before being terminated by heat. This will be followed with a video simulation of restriction enzyme digestion. Okay, so now we are going to start with our restriction enzyme digestion simulation. We are going to start with level 3. And go straight to protocol. First protocol is to set the temperature of the water bath, then set the volume of the P20 micropipette. After that, draw up the reaction buffer into the micropipette. The restriction buffer into the K positive tube. Pipette the restriction buffer into K negative, A positive and A negative tubes. Dispense the other solution into the K positive, K negative, A positive and A negative tubes. Spin the solution tubes. Incubate the solution tubes at 37 degrees Celsius. Store the solution tubes in the freezer. Okay, let's start the simulation. First, set the water bed temperature to 37 degrees Celsius. 37 degrees Celsius. Okay. Next, select the P20 micropipette. Set the volume to 4 microliters. Save. Attach a tip to the micropipette. So we are going to attach a P20 micropipette tip. Okay. Move the rest to the foot tube to the empty tube rack and open it. Restriction buffer. Open it. Draw up 4 microliters of restriction buffer. So let's draw up 4 microliters of restriction buffer here. Okay, and close the tube. Open the K positive tube. Okay, dispense restriction buffer into K positive tube. We are going to close the tube and eject the tip into the trash. Next, we are going to dispense 4 microliters of restriction buffer into the K negative tube. So let's attach a tip and dispense 4 microliters of restriction buffer into K negative tube. So let's dispense into K negative tube. Okay. Then we are going to tip and attach a new tip. Dispense 4 microliters of restriction buffer into the A positive tube. We are going to close this K negative tube and
dispense 4 microliters of A positive, restriction buffer into the A positive tube. So we are going to dispense into it and close the tubes. Eject tip. Dispense 4 microliters of restriction buffer into the A negative tube. So we are going to dispense 4 microliters into A negative tube. Tip as well. Next, dispense 4 microliters of pKan R into the K positive tube. So we are going to put this buffer restriction buffer away and take this out, this buffer, and attach a pipette. Then we are going to dispense 4 microliters of the buffer into the K positive tube. Going to eject the tip and attach a new tip to the micropipette, because we are going to dispense 4 microliters of the same buffer into the K negative tube. So we are going to close these tubes and eject the tip of the micropipette into the trash, because we are going to dispense 4 microliters of PARA into the A positive tubes, and we are going to put this buffer away. Pick this buffer and put it in the empty rack. Attach a new tip to the micropipette and dispense 4 microliters of the buffer into the A positive tube. A positive tube. We are going to close this tube and then eject the tip into the trash. Attach a new tip and dispense the same buffer in 4 microliter volume into the A negative tube. A negative tube. A negative tube. Alright. We are going to eject the tip and then close these buffers. We are going to change the volume of the micropipette, P20 micropipette, to 2 microliters. So 2 microliters. Save volume. Dispense 2 microliters of restriction enzymes into the K positive tube. So we are going to take this out and take the restriction enzyme buffer out. Attach a new tip to the micropipette and dispense 2 microliters of the buffer into K positive tube. Then we are going to eject the tip and attach a new tip, and then the same restriction buff enzyme into the A positive tube. We are going to close this K positive tube and open the A positive tube and dispense the buffer into it, enzyme. Then we are going to close both these tubes and eject the tip. Now we are going to put this restriction enzyme away, because we are going to dispense 2 microliters of distilled water into the A negative tubes. So basically we are just dispensing these distilled waters into the negative tubes, which is A negative and K negative tubes, and we are going to dispense 2 microliters of them. So we are going to A negative tube first, and let's eject it. Eject the tip, attach a new tip, and do the same thing. Dispense 2 microliters of distilled water into the K negative tube. eject the tip, place it at its place, and we are going to put the distilled water away because we are not going to use this anymore. Okay, next we are going to open the microcentrifuge and place the solution tubes into the microcentrifuge. Open and place the samples into the microcentrifuge. We have to make sure that the microcentrifuge is always balanced, which is each tube is opposed by another tube in the direction, and we are going to check the balancing. Microcentrifuge looks balanced and ready, so the microcentrifuge should look like this. Okay, now we are going to close the microcentrifuge and set the microcentrifuge to spin for 10 seconds. Start. And remove the solution tubes. We're going to open the microcentrifuge and remove the solution tubes from the microcentrifuge. Close, and we are going to close this. Then we are going to incubate the solution tubes at 37 degrees Celsius. We are going to open the water bath, which we have set the temperature at 37 degrees Celsius. In the earlier of the video, before starting the other procedures, now we are going to insert the floating solution tube rack into the water bath. We are going to transfer all the samples into the floating tube rack, and we are going to place this tube rack into the water bath and close the water bath. Now we are going to set the water bath timer for 1 hour. Start. So basically we are going to wait until this 1 hour timer on water bath runs out, and we are ready for the last, which is the 9th protocol for this restriction enzyme digestion simulation. Alrighty, we are all set. Close. Next, now we are going to open the freezer and place the samples in the freezer. So we are going to place the samples from the water bath right in the floating tube rack into the freezer and close them both. Next. Okay, so these are the results, actual results and ideal results. And this is the results on the gel electrophoresis. Next section is a reflection, so let's try doing this questions. Why was using restriction enzyme necessary in this simulation? Restriction enzyme is usually used to digest the plasmids in specific locations to isolate areas of interest. What is the purpose of the restriction buffer? The purpose of the restriction buffer is to provide the optimal pH environment for enzyme function. Why is gel electrophoresis used following a restriction digest experiment? It is used to separate and verify the size of the fragments generated by the restriction enzymes. Why is distilled water added to the control K negative and A negative tubes, but not this experimental K positive and A positive tubes? Distilled water is usually added to the control tubes to keep the volumes of the experimental and control tubes consistent, because we are going to microcentrifuge the samples. So we are going to make sure that the tubes are in the same level and consistent. And the final question, why do we use the same two restriction enzymes for both plasmids? we are going to use the same two resistant enzyme for the both plasmids because we are going to ensure that the fragments have complementary sticky ends that can be joined together. Okay, feedback question. How confident do you feel about using the technique you learned? I feel very confident because I feel like I've learned everything that I should know for this restriction enzyme digestion. So this is a summary. In the simulation, I have learned on how to perform a restriction digestion simulation. I have learned on how to micropipette multiple solutions and set up experimental and control samples, which are A positive and K positive for experimental and A negative and K negative for control samples. I've learned on how to balance and operate a microcentrifuge when we place the samples right opposite of each other to make sure the centrifuge is balanced, and I have explained the effect of temperature on enzyme, which you can basically rate at the results area. So end simulation, and that's it. Thank you very much for watching.